Good morning, everyone. This is Sir Franz, and will be our last lesson for anatomy and physiology. And this morning, we will be talking about glucose homeostasis. So, in this lecture, we will be learning how the body controls the amount of glucose that is present in the plasma, which is then circulated throughout the body and will be made available to the peripheral tissues. So we have to remember that glucose is a ready source of energy. So it is the primary energy source by the cells of the body in the fed state. So when we talk of the fed state, we are referring to the body, the human body, after it has been given a meal. So that spans around 1 to 6 hours after eating. Glucose supplies cellular energy when it is broken down. So, when we have something to eat and um, the materials that we have eaten have been digested and have been absorbed by the gastrointestinal system, there is a process by which the glucose that is in the blood enters the cells of the body. And once this glucose has entered the cells of the body, the cell will, in an organized manner, break down the glucose to liberate energy, which is in the form of adenosine triphosphate. So here are the three steps by which that happens. Glycolysis is the first step, and the end product of glycolysis is pyruvate, which is further processed or catabolized to form acetyl coenzyme A. The Krebs cycle then utilizes acetyl coenzyme A to produce reducing equivalents by oxidation of carbon. So you can search for this process through the internet or learn about this more in your biochemistry. But suffice it to say that acetyl coenzyme A has two carbons, and those individual carbons are oxidized. That means that the cells will take away from the carbon electrons, and these electrons are transferred to reducing equivalents, particularly NADH and FADH2. So the Krebs cycle will have to perform those metabolic reactions at the end of the day to produce the reducing equivalents, so that the electron transport chain uses these reducing equivalents and handles the electrons and hydrogen through the mitochondrial membrane to produce adenosine triphosphate. So that means that the glucose that has entered the cell is further broken down to produce adenosine triphosphate. And these are the cellular fates of glucose. Next, this slide shows us glucose absorption in the gastrointestinal tract and splanchnic disposition. When we use the term splanchnic, we are referring to the abdominal viscera or organs, particularly those of the digestive system and the accessory organs such as the liver, the spleen, and the pancreas. So when we say again splanchnic, we are referring to the terminal part of the esophagus, the stomach, the small intestines, the large intestines, and the solid organs such as the liver, the spleen, and the pancreas. So this slide will tell us what happens to glucose once it is already present in the abdominal viscera and how the abdominal viscera processes that glucose. So, first one, the enterocytes or the epithelial cells of the small intestines, no, the cells that are found in the mucosa, will absorb glucose after it is liberated by digestion of polysaccharides. So, yung starch na inyong kinain, you chew that, then swallow, and then that enters the stomach and eventually the duodenum, where in the duodenum, the starch encounters amylase that is secreted by the pancreas, and that will digest the starch into individual glucose units. So the individual glucose units will then encounter the small intestinal cell by which it is absorbed by the small intestinal cell. So glucose from the small intestine cell will then leave the cell and enter the venous drainage of that cell or of the small intestines. And that is illustrated here in the image in the right. As you will see, the stomach has been cut away, and what is seen here would be you have to focus on the middle, the small intestines. No? So that's the duodenum, and you will see here that the duodenum is drained by mesenteric veins that will fuse together to form the portal vein, which directly supplies blood to the liver. So let's connect all of those processes. You have already absorbed the glucose from the small intestines. Pumunta na siya sa mesenteric veins, in the blood that is in the mesenteric veins, into the blood that is present in the portal vein, which will then enter the liver and bathe the liver cells or hepatocytes. So, in that process, you have transferred the glucose from the small intestine to the portal vein and then into the liver. Glucose that spills over will then reach the endocrine pancreas and trigger insulin release. So, in this process, what happens is that the glucose that is in the small intestine will eventually find its way in the liver. And the cells that are in the liver will continuously remove from the blood glucose. So in this way, the liver functions as a gate, no? It functions as a filter for glucose. So as glucose that is in the blood bathes the liver will take glucose from the blood, capture that glucose, and whatever is not captured by the liver will be the one that reaches the systemic circulation. So, from the liver, the hepatic veins, the inferior vena cava, and it goes back to the heart, the right atrium, right ventricle, and then the lungs, the left atrium, left ventricle, aorta, and systemic circulation. And then from the aorta, eventually to the pancreatic artery, which will then bathe the pancreas with now glucose-rich plasma. And in response to that glucose-rich plasma, the pancreas will then release insulin, which we will discuss um, in the next few slides. So again, we have here how the abdominal viscera, particularly the small intestines, process the glucose, transfers it to the liver captures the glucose, the excess glucose will spill over into the blood, reaches the pancreas, which in response will secrete insulin. So this next slide will tell us how the liver captures the glucose. So in the first slide, we talked about how glucose is broken down into pyruvate via glycolysis. So the first step by which glucose is broken down through glycolysis into pyruvate is by attaching a phosphate molecule into glucose. So the enzyme that attaches that phosphate molecule into glucose is hexokinase. So hexokinase is present in all the cells of the body. Okay? So the liver has a particular type of hexokinase that we call glucokinase. So, ibig sabihin nun, glucokinase is an isoform of hexokinase. So, that also means that glucokinase is an isoenzyme of hexokinase. So, let's first look at this graph. Hexokinase is labeled in red. The x-axis of this graph is glucose concentration, and the y-axis is the maximal effect of the enzyme. So let's take a look first at the red line. You will observe that the maximal effect of hexokinase is observed at a lower amount of glucose. So that means even at a glucose concentration of 100, that is relatively low, hexokinase will already be active. Hexokinase will already attach phosphate to glucose molecules even at a lower concentration of 100. But what's special with glucokinase, which is our isoenzyme found in the liver, is at a glucose concentration of 100, it has low activity, and its maximal activity will only be reached at a level of 200 to 200 plus milligrams of glucose per 100 ml. So what does that mean? Glucokinase will only be active at higher levels of glucose, which is what happens after you have eaten your meal. So, what does that tell us? Glucokinase, which is found in the liver, will only be active and will only trap glucose whenever the blood that reaches the liver has high levels of glucose, which only happens after a meal. So, once you have eaten your meal, the duodenum has digested, processed the starch to produce glucose. The duodenum and the small intestines absorbs that glucose and creates blood that has high levels of glucose in the portal vein. The blood that is in the portal vein, rich in glucose, bathes the liver. The liver will then be actively trapping the glucose because it is only at the time of rich glucose in the portal blood that glucokinase is active. Okay, so once you have finished the absorptive phase in the small intestines, the levels of glucose in the portal blood will continuously go down. And when that level goes down, glucokinase will switch off because the levels in the portal blood are low already. So in that instance, in the absorptive phase, in the fed state, glucokinase and the liver will trap glucose. So again, whatever is not trapped by the liver will spill over into the blood, which will eventually reach and communicate with the pancreas, which is our next slide. So this is the endocrine pancreas. You have to understand that there are two types of tissues in the pancreas, exocrine and endocrine. Pag sinabi nating exocrine, we are referring to the pancreatic acini, which you will see here in the right image. This pancreatic acini, and particularly the cells here, the acinar cells, will create or synthesize your digestive enzymes. These digestive enzymes are in the form of amylase, which will digest starch, trypsin, and chymotrypsin, which will digest proteins, and lipase, which will digest fat. So these digestive enzymes that are secreted into the duodenum come from the exocrine pancreas, which are made up of pancreatic acini. But here in our discussion in endocrinology, we are more concerned with the endocrine pancreas, which is a particular set of tissues which in general we call the islet of Langerhans. So here in the image in the right, the islet of Langerhans is in the middle, no? And that is composed of beta cells, which are the major population um, of cells in the islet. You also have the alpha cell and the delta cell. So these cells make up the islet of Langerhans. So the islet of Langerhans contains many cell types, and each cell here will secrete a specific hormone. For example, the beta cell, which makes up most of the islet of Langerhans, will synthesize insulin. The alpha cell will synthesize glucagon. So the question here is, how do these individual cells secrete their hormones in response to the levels of glucose in the blood? So that is what is shown here in this slide. First, let's look at this image in the left portion. The stimulus is an increase in blood glucose, which happens when the liver spills over the glucose that is in the portal blood. So that spilled over glucose reaches the systemic circulation, which at the end of the day bathes the pancreas through the pancreatic artery and bathes the islets of Langerhans with extracellular fluid that has high levels of glucose. So, this high level of glucose will then be encountered by the beta cell, which is shown here. So, this is the beta cell. The high levels of glucose, the glucose in particular, will enter the beta cell through the glucose transporter type 2 or GLUT2. Glucose will then undergo glycolysis, so it will be acted upon by glucokinase, which again is active only at high levels of glucose. Glucose, through the action of glucokinase, magdadagdag siya ng phosphate. It will then form glucose 6-phosphate. Glucose 6-phosphate will then undergo glycolysis to produce pyruvate. It will then be converted to acetyl coenzyme A. Acetyl coenzyme A will then be catered by the Krebs cycle to produce reducing equivalents, and the reducing equivalents will then be taken up by the electron transport chain, processed through oxidative phosphorylation to produce high levels of adenosine triphosphate or high ATP. Once the pancreatic beta cell has already created lots of ATP, that will then close the potassium channel, excuse me, which in this instance, causes potassium to stay inside the cell. So we have to understand again or remember that potassium has high levels intracellularly compared to the extracellular environment. So if you close the potassium channel in response to high levels of ATP, potassium will build up or increase in concentration intracellularly. Since potassium is a cation, that will make the interior of the beta cell progressively more positive, and that will lead to depolarization or a more positive intracellular environment. Once depolarization has already occurred, that will lead to the opening of calcium channels. Again, remember your nervous system physiology. The calcium channels are voltage gated or they are voltage sensitive, which means they reflect the positivity or the negativity of the interior of the cell. So since your calcium channel is voltage gated, it will respond to depolarization, which is the creation of a more positive internal milieu. So that depolarization again is caused by the closing of the potassium channels and the buildup of potassium. So the calcium channels open. And you have to understand again that calcium is present in the extracellular environment. So upon opening of the calcium channels, calcium will enter the cell from the outside. So this will lead to an increase in the amount of calcium inside the cell. And once the interior of the cell has lots of calcium, that will cause the vesicles that contain insulin, represented here as the purple circles. So that will cause the vesicles to fuse with the cell membrane of the beta cell, leading to the release of insulin. So through this process, in response to high glucose concentrations, the beta cell releases insulin. Insulin will then um, enter the circulation and exert its action in the cells which carry its receptor. So you have to understand that insulin is the major anabolic hormone. If we use the word anabolic, we refer to chemical reactions that build up, that create larger molecules from smaller molecules. So that is what is referred to by the term anabolism. No? So insulin secreted by the pancreas serves to coordinate a shift in metabolism. What is this shift? This involves the prioritization of glucose for energy production. Instead of utilizing fats and proteins to produce energy, if insulin is secreted, the cells of the body will then shift their attention, change their focus from utilizing fats and proteins to utilizing glucose. Instead of breaking down fats and proteins to produce energy, they will now use glucose to produce energy, which is what we discussed in the first slide. Insulin facilitates anabolism. So again, anabolism is the making of large molecules from smaller component molecules or building blocks. This include triglyceride synthesis, which is the making of fat from 3 units of fatty acids and 1 unit of glycerol. You combine 1 glycerol and 3 fatty acids to form triglycerides. Protein synthesis, that's also stimulated by insulin. So you combine individual amino acids which you have taken up from the food to produce proteins. And insulin also stimulates glycogen synthesis, which means that the excess glucose that has not been trapped by the liver will be taken up, particularly by muscle cells, and combined to each other to form glycogen. So you can think of glycogen as the storage form of glucose in the human body. That is very special because the liver, aside from removing glucose from the blood and utilizing that glucose through glycolysis, the liver will also store that glucose in the form of glycogen. Okay? Insulin will also inhibit catabolism of tissue stores. So, if insulin has already been released, insulin will prevent the degradation of glycogen to release glucose. It will prevent the degradation of protein to release amino acids. It will prevent the degradation of triglycerides to release fatty acids. So, in that way, you are protecting your tissue stores, which is sensical or sound because you are able to eat and therefore you don't have to utilize your tissue stores. Insulin will also, in the same way or idea, prevent lipolysis or the breakdown of fat. Insulin also, which is what is the basis of the utilization of insulin in the treatment of diabetes mellitus, insulin also lowers blood glucose by causing its absorption into muscle and fat. So, insulin, by way of its intracellular signaling, will cause muscle cells to remove glucose from the blood by transferring it to its interior, or in simpler terms, absorption. The muscle cells will absorb glucose from the blood. Fat cells will also absorb glucose from the blood. So, by absorbing the glucose, what is reflected in the plasma is a lowering of glucose. So, how does insulin work? That is through this image right here. So, insulin is that turquoise colored shape. No? So, insulin will bind to the insulin receptor. The insulin receptor has two portions, an alpha subunit, which is extracellular, and that is connected via disulfide bridges to the beta subunit, which is a trans membrane protein, which has an intracellular component. So again, the insulin receptor has two subunits, an alpha or extracellular subunit and a beta subunit, which has a transmembrane property and therefore has an intracellular component. Again, insulin binds to the insulin receptor. The insulin receptor, upon binding of insulin, will then activate tyrosine kinase. So, this enzyme attaches phosphate groups to lots of proteins through the tyrosine amino acid component of those proteins. So again, The tyrosine kinase is an enzyme that attaches phosphate to tyrosine residues of particular proteins inside the cell. So these proteins are what we call IRS, insulin receptor substrates. So before the binding of insulin, noong wala pang insulin, yung IRS, lumulutang lang siya in the cytoplasm of the peripheral cell. But upon binding of insulin to its receptor, tyrosine kinase becomes activated. And yung mga lumulutang na IRS will then receive phosphate groups via the action of tyrosine kinase, which attaches the phosphate to a tyrosine amino acid present in the protein, which is your IRS. So once these proteins have already been phosphorylated, they become active. So once these proteins are already active, what do? These proteins are actually enzymes that facilitate particular steps in metabolism. So for example, if the enzyme that has been phosphorylated is involved in protein synthesis, that will manifest as an increase in the rate and efficiency of protein synthesis. And that is how insulin facilitates protein synthesis. If the enzyme activated by phosphorylation is involved in the synthesis of fat, then that is how insulin facilitates fat synthesis. If the enzyme involved is a key enzyme in glycogen synthesis, then that is how insulin facilitates glycogen synthesis or glycogenesis. If the enzyme involved is important for growth of the cell or of the tissue type, then that is how insulin causes growth and gene expression. The last effect of insulin, again, as we have discussed a while ago, is the lowering of plasma glucose. And that is through the phosphorylation of vesicles that contain glucose transporters. So upon phosphorylation of these vesicles that contain glucose transporters, glucose is upregulated or inserted into the cell membrane of muscle cells and fat cells. If the glucose transporter is already active and available in the cell membrane, then that will create a pore upon which glucose from the extracellular fluid is able to enter and pass through to reach the intracellular fluid. And that is how glucose that is in the plasma, in the blood, enters the muscle cell, because glucose transporters have already been made available. So in that effect, as glucose enters the muscle cell, as glucose enters the fat cell, glucose in the blood goes down. And this particular transporter which responds to insulin is your GLUT4 or glucose transporter type 4. So, through all of those discussions, we have understood what happens to the organism and to the cells of the organism or the human during the fed state. Now, let's discuss what happens during the fasting state. Fasting technically means that period in time where the organism has nothing to eat. So what happens during the fasting state? The counter-regulatory pathways are activated. And the goals of the counter-regulatory pathways would be to create a shift in metabolism so that one prevents hypoglycemia or a very low level of glucose in the blood, which is detrimental to the organism. So, what happens here? When glucose flux from the intestine decreases because the organism did not eat or is not able to eat, blood glucose is maintained by the release of stored glucose in the liver and the synthesis of new glucose from amino acids, lactate, and pyruvate. So you have to contrast this with what happened during the fed state. During the fed state, glucose that has reached the portal vein bathes the liver, and in that instance, the liver captures the glucose and stores it for future use. Now, this time, when you don't have something to eat, that's the time that the glucose that has been captured by the liver in the form of glycogen is released so that you prevent the blood um, concentration of glucose from falling down. Okay, so that is better represented in the image we have here in the right. So in the center of this image, you see a balance or a scale. And the goal of the counter-regulatory pathways and insulin would be to keep this scale in balance. Let's start first with the left side of the image where blood glucose is high. We have already discussed that the liver captures the glucose and the excess glucose is detected by the pancreas, upon which it secretes insulin. And that insulin will cause an increase in glucose uptake by the fat cell and the muscle cell. And that insulin stimulates again the liver through glycogenesis or the formation of glycogen, which in effect captures the glucose. And through the liver, the insulin will also decrease the amount of gluconeogenesis or the formation of glucose. If you are able to take up glucose, if you are able to capture glucose in the form of glycogenesis, and if you are able to prevent the formation of glucose through gluconeogenesis, that will reflect as lowering of blood glucose to normal levels. So, in summary, your stress is an increase in blood glucose through a meal. And in response to that stimulus or stress, insulin decreases blood glucose. So, you kept the balance or the scale from being tilted towards high glucose. So you kept the glucose in control. What then happens when you are in the fasting state and you don't have something to eat? That is shown here in the right side of the image, where at night you sleep and you don't eat. Blood glucose falls, and the glucose that baits the liver will go down. The glucose that baits the pancreatic islets of Langerhans goes down. And in response to a lowering of blood glucose, the alpha cells of the islets of Langerhans secrete glucagon. Glucagon then travels through the blood, much like how insulin travels through the blood. Glucagon then encounters the liver, where the receptors for glucagon are present. The liver cell or the hepatocyte, in response to glucagon, will then facilitate glycogenolysis or the breakdown of glycogen to release glucose. The liver, in response to glucagon and epinephrine in particular, will then facilitate gluconeogenesis, which is the utilization of amino acids, lactate, and pyruvate to form glucose. And these two processes will then lead to the liver releasing glucose, which will then combat the lowering of blood glucose. So, the liver, through glycogenolysis and gluconeogenesis, will increase the output of glucose to cause a normalization of the balance of glucose. So, in the upper image, you prevented the tilt towards hyperglycemia. And in the lower half of the image, during the fasting state, you prevented the tilt of the balance towards hypoglycemia. So, in effect, through these counter regulatory pathways, you have always kept that balance in level. Now let's discuss the counter-regulatory hormones, which are technically hormones that oppose the action of insulin. Glucagon in the liver will stimulate glycogenolysis. Epinephrine, by its action in the liver and in the fat cell, will stimulate lipolysis so that you release free fatty acids, which will then become the alternative energy source of the body in states of the fasting, in the fasting state. Glycogenolysis is also stimulated by epinephrine. Gluconeogenesis, or the formation of glucose, is also facilitated by epinephrine. Glycogenolysis again and gluconeogenesis will cause an increase in the output of glucose by the liver. Growth hormone is also released in the fasting state, and the effect of growth hormone will be insulin resistance. So, in the fasting state, there is insulin resistance, so that the muscle and the fat cell will not take up glucose. This prevents the action of insulin so that glucose remains in the blood and is spared for the brain, so that glucose in the blood will stop from going down because you have lower activity of insulin. Growth hormone also facilitates lipolysis. Again, the goal is to release free fatty acids, which will serve as an alternative energy source. Growth hormone will also stimulate gluconeogenesis. It will stimulate glycogenolysis. But what's special with growth hormone is that it keeps proteins in mind, so that even in the fasting state, you have an increase in protein synthesis and an inhibition of protein breakdown or proteolysis, so that your lean muscle mass or your lean protein mass does not go down. You have to understand that the structures of the organs of the body are made up of protein. So in the fasting state, there has to be a way that the human body prevents injury to its own organs so that it is not sacrificed to produce energy. And that is through stimulating protein synthesis and inhibiting proteolysis. Can you just imagine that if you don't have something to eat, there is nothing that stops the body from breaking down the protein that is found in the heart. So, if there is no growth hormone, which slows down protein breakdown, the heart will start to fail because its structural proteins are broken down for energy. So that is the role of growth hormone, to protect the tissues of the body from being broken down to liberate energy. The last one would be cortisol, which stimulates the production of enzymes for catabolic pathways and also stimulates gluconeogenesis. Our last slide would be the application of these physiologic principles into a disorder. And the best disorder to talk about this or to reflect these principles would be diabetes mellitus. So diabetes mellitus is actually any disorder that manifests as hyperglycemia. Hyper means high, glyc means glucose, and emia means blood. So you have high levels of glucose in the blood. So any disorder that manifests as a high level of glucose in the blood is actually diabetes mellitus. Diabetes mellitus creates a disastrous scenario because chronic elevations of blood sugar will lead to injury to the nerves and the blood vessels. So, persons with diabetes mellitus do not die of an elevated blood glucose per se, but die of the complications of diabetes mellitus, such as injury to the kidneys, injury to the blood vessels, which predisposes the patient or individual from suffering from heart attacks or strokes. So these are the usual pathways of death in diabetes mellitus. So in general, via pathogenesis, we have two types of diabetes mellitus. Type 1, which is type 1 DM, also known as insulin-dependent diabetes mellitus or diabetes mellitus of the young, is due to autoimmune destruction of beta cells. So, the immune system of the individual attacks the beta cells, and the beta cells in the islets of Langerhans die, and because of that, insulin secretion is absent. The second type of diabetes mellitus via pathogenesis is type 2 DM, where there is enough insulin secretion by the beta cell, but the peripheral cells, particularly the muscle and the fat cell, are resistant to the action of insulin. Their insulin receptors do not create insulin effects, and therefore, at the end of the day, glucose is not able to enter the muscle and the fat cell, and that reflects as high levels of glucose in the blood, diabetes mellitus. So, what is shown here in the image in the right is um, the pathophysiology of type 2 diabetes mellitus. There are many dysfunctional processes that all lead to hyperglycemia. The first one is an increase in glucose influx. In patients with type 2 diabetes mellitus, their gastrointestinal tract are abnormally more efficient in absorbing glucose than a healthy individual. So that if you take in the same amount of glucose via food, patients with type 2 diabetes mellitus will be able to absorb more glucose than usual. Patients with type 2 diabetes mellitus have an exaggerated release of glucagon, and that spells disaster because that leads to highly active glycogenolysis and gluconeogenesis, which will increase the output of glucose into the blood. Patients with type 2 diabetes mellitus have relatively low insulin secretion compared to patients which have normal um, pancreatic physiology. patients with type 2 DM have abnormal neurotransmitter release. The amounts of acetylcholine that is released is lower, and the amounts of epinephrine that is released is higher, so that all of these will shift metabolism towards the catabolic phase. The muscles and the fat cells have low response to insulin activity, and so they have decreased peripheral glucose uptake. They cannot take glucose from the blood, and therefore glucose stays in the blood and manifests as hyperglycemia. The kidneys in normal persons have the ability to secrete glucose into the urine, and they also have a particular ability to reabsorb glucose. In patients with type 2 diabetes mellitus, their nephrons are not able to eliminate into the urine glucose as efficiently. But what's weird with them is that they also have a higher efficiency of reabsorbing glucose that should have been urinated into the urine, but then finds its way back again into the blood, and that will lead to hyperglycemia. And lastly, patients with type 2 diabetes mellitus are able to output um, glucose from the liver more efficiently than normal persons, so that their livers are unable to capture glucose, and if they do um, store glucose, they release that glucose more freely, and they create new glucose molecules through gluconeogenesis more avidly. So that will lead to hyperglycemia. So all of these processes will synergistically act to produce hyperglycemia, which again becomes chronic in diabetes mellitus and eventually injures nerves and blood vessels, which will increase complication rates, particularly of stroke and heart attack, which will cause the individual to die. So that's it for our discussion of homeostasis of glucose. I hope you learned a lot. And please do not forget to read your book and read scientific articles. Thank you.